Hello everyone and welcome to Filtering Out the Noise. In this webinar, kindly sponsored by Zymo Fertility, we will uncover the latest methods and best practices in conventional insemination and fertilization techniques. Our esteemed panel of experts will share invaluable insights and tips from the lab, providing a comprehensive understanding of the procedures involved. Topics will include reviewing cutting-edge techniques, behind-the-scenes access to state-of-the-art labs, and uncovering the latest research on the intricate molecular mechanisms that drive the basis of fertilization. I'm very excited to be co-hosting this evening with the multi-talented Neringa, who is a trainee embryologist, STEM ambassador, and scientific writer. Currently, she's completing the STP training program and training as an embryologist at Care Fertility in Bath.
Thank you, Kimberly. Kimberly is a senior clinical embryologist and lab director at Fertility Center Hamburg, which was the first outpatient IVF center and is one of the largest IVF clinics in Germany. Now, allow me to introduce our panelists. Dr. McCaffrey is an active member of many national and international organizations, including ASRM, the Fertility Preservation Special Interest Group, ESHRI, American College of Embryology, and New York Metropolitan Embryology Society, and has served on the board of SARD. Dr. McCaffrey has presented at national and international meetings, as well as authored and co-authored many peer-reviewed publications, including several book chapters on various topics related to IVF. Our second panelist, Professor Gianpiero Palermo, is a renowned figure in the field of reproductive medicine and andrology. He is the developer of the revolutionary intracytoplasmic sperm injection procedure in 1991. Professor Palermo is the director of assisted fertilization and andrology at the Center of Reproductive Medicine and Infertility, and an associate professor at the Well Medical College of Cornell University. But let's hear from Dr. McCaffrey.
Thank you, Neringa. And thank you everyone for joining. Can you hear me okay?
Yeah, we can.
Great. So I have the pleasure of introducing this very interesting topic. Again, I just want to make sure that you can see my screen. You can see?
Yes, we can.
Okay. Sorry, just back. So I'm introducing this topic of fertilization, what matters, and filtering out the noise to the speakers who I'm really excited to hear what they have to say. So I won't take up much of the time, but I just wanted to introduce the topic. And I think, you know, basically what we're all aiming to do is to find, get a really successful outcome in choosing the right sperm and the right egg, so that hopefully we create a beautiful embryo, which eventually becomes a live birth for our patients. So the question is, how do we do that? So at NYU, where I've worked for the last 20 plus years, we have always tried to practice where we choose patients to do ICSI for those male factor patients rather than do it across the board. So our use of ICSI has been lower than the national average at all times. And, you know, we strive to constantly try and find ways to identify the patients that need ICSI versus those that can benefit from ICSI. So we feel that, you know, it really is the lab responsibility to find out, you know, how to identify the best cells, how to handle the samples in the lab to ensure integrity is preserved as much as possible, and how to ensure best practices in the lab from a safety as well as medical point of view. And lab practices certainly differ across the world and across the country. Many labs rely heavily on ICSI, and in those cases, you don't need a high number of sperm, but you certainly want to choose the best sperm to use. The debate on INSEM versus ICSI is a constant debate. Again, using an invasive procedure for insemination versus eliminating or at least reducing as much as possible the chance of a failed fertilization. However, despite this ongoing debate, it's clear that the incidence of ICSI for non-male factor diagnosis is increasing across the USA and the world, even at this point. So as we strive to identify and find the best sperm to use for insemination or for ICSI, there are a variety of ways to process the sperm, and I'm just listing the main ones here that we're all familiar with: gradient layer and centrifugation, simple wash if there's very few sperm available, swim up if you have a very good sample and progressive motile sperm, and then more in recent years, the separation devices, for instance, the Zymot, which we've all become very familiar with. We use extensively now. Again, our procedures, this is just a very brief summary of our results at NYU. We use Zymot very extensively now for sperm separation. We've used it for selecting sperm for ICSI for about the last 2 years, almost exclusively. And now for insemination for the past year, we've moved to using Zymot and swim up mostly for separating sperm. So I just summarized here the results from our Zymot preparation for conventional insemination cases, and just to give you an idea, we like it because we end up with a very high percentage of motile cells in the final prep. And when we've done insemination with the Zymot prep sperm, we certainly reach fertilization rates that are comparable with any other method of sperm prep as well. So, as I said, we pretty much have adopted using the Zymot separation device in our lab for samples definitely for ICSI, and a lot of it now for insemination. Of course, if the sample is a good sample and we can do swim up, then we also use swim up. But we like the Zymot separation device, and it's been very successful because, first of all, most importantly, after testing and the initial modification of the protocol in which the last wash was included in the process, we have data to show that it works. So that's the most important thing. It's certainly, once you get used to it, it's very simple to use. The final specimen is very clean with a high % of motile cells obtained. We find in a busy lab like ours, it saves time in the lab for processing, probably at least about 20 minutes per sample, which at the end of the day certainly adds up. It reduces centrifugation steps compared to a prep like gradient prep, less handling of the sample, and it streamlines procedures in our busy lab for sure. So, and that's pretty much the introduction. I'm excited to hear the rest of the speakers and to learn more today and how we can improve. But thank you for allowing me to speak and to introduce the topic.
Thank you very much, Caroline. That was an amazing overview. Thank you. To everyone, please, when you have questions, place them in the Q&A box, not in the chat. But seeing as I'm one of the moderators, I have the privilege of getting to ask the first question. So Caroline, when you decide on whether to do IVF or ICSI, does morphology play a role in that decision?
No, actually, very, very little role. I mean, of course, if it's a sample that's grossly abnormal and has, missing like an acrosome, it certainly does play a role. But otherwise, if we have a good count and good motility, we don't worry about, the strict Kruger morphology or any of that. So no, pretty much in our lab, we don't take morphology into consideration.
That's amazing. That's certainly something very, but new to think about. I think it's great that you're not placing such a big importance on morphology. So thank you very much for that. I'm sure we'll have lots of more questions later on, but for now I will introduce the next talk. The next talk that we will hear is recorded and it will set the scene. It's given by Xavier Gonzalez, who is a senior clinical embryologist and laboratory manager at ARIA in London. Xavier is a lecturer for the postgraduate degrees in women's health, prenatal genetics, and fetal medicine at University College London too.
Thank you so much for inviting me tonight. Good evening, everyone. My name is Xavier, and today I will be sharing my experience on improvements in the context of sperm preparation. Improvement is a word that resonates with all of us, whether it's about streamlining a process, achieving greater efficiency, or ultimately attaining better outcomes. So just before we start, a quick word about myself. I'm a senior embryologist and lab manager currently working in London, and I'm also a PhD researcher at University College London. At ARIA, in our laboratories, we are highly dedicated to research and development, constantly integrating new technologies into our workflow. We use AI for gamete and embryo selection. We are working on implementing time-lapse imaging to optimize non-invasive PGT, and we have experience with single sperm vitrification, which I believe will be something explored tonight by one of the other speakers. In collaboration with UCL, we continue to run a global database where we are looking into patient perspectives and clinical outcomes of new non-euploid embryo transfers. Now, most of us are familiar with the concept of semen analysis, and that a typical ejaculate could contain hundreds of millions of sperm cells. Unlike eggs, working with sperm offers an opportunity for us as embryologists to make a difference through preparation and selection methods. The challenge arises when we have multiple sperm with similar appearance, motility, shape, morphology. So it becomes a bit tricky to select those sperm. So how can we select the best sperm for successful fertilization and subsequent development? To understand the significance of sperm preparation, let's first consider very briefly what happens in vivo. So basically, sperm undergo rheotaxis, moving upwards from the vagina, which leaves some sperm behind, obviously, in the uterus. Thermotaxis and chemotaxis guide sperm towards the fallopian tubes, where only a few and ultimately one will reach the egg. So why do we need to prepare sperm sample in the IVF lab? So sperm obtained immediately after ejaculation cannot fertilize eggs, and we must separate sperm from seminal plasma and induce processes of capacitation, hyperactivation, and membrane changes. So there's different concepts when we work with sperm in the lab. We can talk about preparation methods such as density gradient, swim up, microfluidics, MAX. And then once the sample gets prepared, we can select that sperm, and that could be by the means of ICSI, PICSI, or using AI as a guide. However, the problem is that we need to acknowledge some challenges. One of them would be the heterogeneity of the sample. Not all samples are the same, or similar sperm won't behave in the same manner. As well, thinking about the bigger picture, we need to include some analysis about the cost of those procedures and the evidence, and how much of that cost and evidence then gets passed on to the patient. And obviously, we need to protect those cells. So keeping damage to the lowest would be obviously the preference when we are preparing and selecting sperm. So when we started our work at ARIA, density gradient preparation was the standard practice for all of our samples. But after a while, we looked at optimizing the pathway, and that's when we considered microfluidic devices. We did what we call an FMEA analysis, which is basically some research into mapping the different steps and potential sources of errors. And we compared the density gradient preparation, the conventional method that most labs use nowadays, with the use of the Zyme, which is a device that I believe many of you might be familiar with. So when we looked into an ICSI sample preparation with density gradients. In our protocols, that was sort of put together into 6 steps, which got reduced to 2 basically when we were looking into the macrofluidic device. And when we were looking into how many things could potentially go wrong, we saw that once we introduced the Zymod, 40% of those points of failure, of those potential mistakes, were eliminated, which is great. So ultimately, we were doing the same thing with less steps and less risk. But ultimately, how this change in practice that we wanted to do will translate into differential outcome for the patient. So as I said, this is going to be some internal and published data, but just to give some context. So before using the Zymod, we're using density gradients. And here in this table, I'm comparing some laboratory KPIs, and I'm looking at before Zymod, when we started doing the Zymod with a protocol of 30 minutes incubation, and then a protocol that we adapted of 10 minutes incubation. And these, I'm looking just at ICSI samples. Just for everyone to get an idea, this is around 1,200 cycles worth of data, where 50% of them were ICSI. So we saw consistent KPIs when it comes to ICSI for senior embryology. So we saw same number of fertilization, blastulation, and utilization. And the numbers were not big enough for us to see statistically significances. But it was nice to see that even if we were changing the protocol of preparing sperm sample, the outcome was not compromised when it came to number of embryos that the patient would generate at the end of the cycle. So obviously, we wanted to look in a little bit more in depth, and since there's a lot of information about Zymod and improving that health of the sperm, we were interested into looking into the PGT cohort. So when we were doing genetic testing, are we getting more embryos that are genetically normal? And the graph here that you see at the bottom left, you can see the different age groups, and we see in 3 different colors the different preparation methods. And we could see perhaps some marginal gains in terms of euploidy rate across all groups. The non-statistically significant, probably due to the sample size, but sometimes it's nice to see a trend to start with. And obviously, we might be getting slightly, perhaps 1 euploid embryo more for some age groups, but how does that euploid embryo behave when we transfer? What's that clinical pregnancy rate? So we saw that across the 3 protocols, the performance of that euploid embryo was not compromised. So ultimately, what we decided is to change our practice of, for ICSI samples, we would just do 10 minutes in the Zymod and then recover the supernatant. With conventional IVF, we do use the Zymod, and we use a protocol of 30 minutes just to make sure that there's enough sperm going through the membrane, and then 5-minute wash. And then for IUIs, we do just a 30 minutes incubation. So it was quite positive to see consistent KPIs and some perhaps some trends in the PGT group. And the take home from this method is that we're doing the similar job with less steps, there's less risks, it standardizes the preparation, and it really does save time on a daily basis without compromising the outcome. So just a bit of an introduction of what's to come in the following slides. Just to give an overview that at ARVI we offer like a male fertility assessment, where we do look at semen concentration, motility, and morphology as standard semen analysis, which includes then DNA fragmentation and hyaluronan binding. And based on that, we decide what treatment the patient would have. So if everything comes up normal, conventional IVF would be the desired insemination technique. We run a culture as well on the sample, and if that was to be positive, or there's something growing there that is resistant to the antibiotic that the culture media has, we would then do ICSI. And of course, in cases of male factor, we would be considering a single sperm vitrification if we really have a severe cryptozoospermia, and do ICSI afterwards. when there's high DNA fragmentation or the maturity of the sperm falls outside the desired range, we would do ICSI or PIMSI, which is a combination of PICSI and IMSI, especially in those patients that have a history of miscarriage. And a brief note on single sperm vitrification. It's a technique that we have consolidated internally. Our internal data in terms of recovery and motility, we would expect to see up to 96% of the recovery rate. So we would tend to find the sperm that we freeze in these single drops, and then almost half of them, or just below half of them, would still be motile after these single sperm freeze. And of course, this sperm is extremely precious, so we have built-in polarized system in our microscope. So that's the desired method of ICSI. We would do spindle location ICSI just to maximize the chances for this couple. And now talking a little bit more about that ICSI and PIMSI. So at ARIA, we use AI when we do ICSIs, not as a selection method, but it's just to have a constant, I would say, second opinion live. So it does improve the consistency of our KPIs. And we've seen for junior members of the teams a reduced learning curve when it comes to for them to get trained on hunting good sperm. And the software that we use specifically, you might be familiar with, is provided by IVF 2.0. It's called the SID, and we use that quite a lot for a research and development perspective. I will be sharing a couple of projects that will help you understand what are we doing internally here with AI and ICSI. When it comes to PIMSI, we probably don't do much of it. I would say it would fall around less than 5% of our patients. So, and the idea of PIMSI is that those sperm that show the ability to bind the hyaluronan, in theory, is supposed to be of higher competence, perhaps more mature, better genetic integrity. And interestingly, we combine in our male fertility assessment the DNA fragmentation and the HBA binding test together, because there's a really nice paper 2016 showing that almost half of the infertile male that show normal DNA fragmentation, the HBA testing was off from the threshold. So we thought it was important to always consider these two tests together. And then PIMSI comes from combining these PICSI and IMSI as both independently have provided some evidence of reducing the chances of miscarriage. So just a note on the using artificial intelligence to assess this single this sperm that we will be selecting by ICSI. We collaborated with a multicentric study run by IVF 2.0, and the data was presented at ESHRE 2022. And this was a really interesting study where we were independently selecting the sperm with the software running on the background, but we were not selecting based on the software. And we saw that different clinics across the globe, we all select sperm in a different way. So there was not much consistent. There was some partial consistency, but it was not exactly 100%, even if we compare senior with senior embryologists. So there were some parameters that were associated with higher fertilization and blastulation. But it was very interesting to see that a program such as this could standardize a technique where we all think that we're selecting the sperm the best way. So, and now thinking back into this AI sperm selection, is that sort of the golden grail? Is going to be the ultimate selection? So we wanted to see whether the selection itself was similar when we were selecting sperm ourselves and embryologists. We did a group of selecting sperm, the embryologists, a negative selection group of sperm that we would never select. And then we selected sperm based on the AI. And we wanted to see whether these sperm selected in these 3 different categories show higher proportion of hyaluronan binding, you know. So we want to see if who was better at selecting mature sperm, even if that's something that visually you cannot see that property. So interesting data that we saw is that, as expected, the seed scoring, which is the AI software, was very similar to the selection that the embryologists would do for ICSI. It is a system that, after all, is trained by embryologists, so we would expect to see similarity. So in this graph, you would be able to see on the left vertical, you'll see all the motility parameters that the software would be registering and analyzing. And in different colors in the graph, you would see the embryologist positive selection. So that sperm that the embryologist would choose for an ICSI, the sperm that would not be chosen, and ultimately the sperm that the seed software would be selecting as A, top quality. So yeah, we can really see an alignment of embryologist positive selection and the seed. The thing is that when then we said, okay, let's analyze the ones that pass and fail the test in terms of do they show binding properties or not, we saw that there was no motility parameter analyzed by the seed that was very differential between that sperm that would show binding from the one that did not. And this is irrespective of the selection group, irrespective if it was selected by the embryologist, positive selection, negative selection, or by the seed. And overall, what we saw in this very pilot study was that the embryologist selection was resulting in at a higher percentage of mature sperm selected. And this could be for many reasons, but the one that perhaps was more obvious is that the seed, the software version that we were using at that time, did not include morphology parameters, so which is something that the embryologist would look beyond the motility parameters. So these results are using seed version 1. I believe now it's there's a second version from the seed, so it would be interesting to look into the data again and see if there will be any differences when it came to selecting sperm that were mature as per the binding test. So of course, we're doing all these things in the lab, and we need to make the patients aware that we are doing these things, because it's ultimately about patient experience. We're here, our work is all for the patients. So how we envision this route would be, there's a phase of visible to the patient. For example, the patient comes into the production room using their fingerprint, they can identify themselves. And of course, at the end of the process, what the patient sees is the success rate, and that might be increased number of euploid embryos. It could be increased clinical pregnancy rate or live birth rate. But the patient needs to as well understand that in the lab we're doing something different. So when we get the samples, we get a CASA system that would analyze the sperm automatically, and we will have the zymote preparation that would bring standardization and consistent preparation method. And then once that sperm gets prepared, then we go into selection, and the patient is at ease that we're using that AI as a second opinion consistently. And we make that decision on doing PIMSI based on really the evidence on the sample on the day, where we would be running a hyaluronan binding test as well. So it gives an idea that to the patient that even if we're doing every day the same thing, we're looking into all the little puzzles, all the pieces from the puzzle, which enhance patient experience ultimately. So in closing, we recognize that IVF is a dynamic and ever-evolving field. Digitization has firmly established its place in our daily practice, and it's here to stay. Moreover, automation in IVF is not just on the horizon, it's imminent, promising further advancements in precision, consistency, and efficiency are just around the corner. And in this era of constant progress, let's us also be mindful of our environmental impact. As we embrace automation and digitization, it's essential to consider greener options and sustainable practices. And of course, we are scientists. We need to improve what we do every day. So we always need to embrace change, but never losing focus on that main goal that brings us all together, which is helping our patients. Thank you so much for listening.
Now, our next speaker, Chaya Rothschild, began working in ART laboratory in 2015 and joined Maze Men's Health in 2019 to oversee the implementation of extended sperm search and microfreeze procedure. She and her team collaborate with 30 IVF centers throughout the US and Europe to use their unique experience to address severe cases of male factor infertility.
Hi, everyone. If I had known that Xavier was going to set me up so well, I could have skipped the first few slides I have for you here. As Neringa just mentioned, I've spent the last few years working in a urology-based andrology lab. So I really spend my time looking at the bad sperm, the terrible cases that that are my favorite topic to talk about, but most other embryologists' least favorite topic. My department was developed to really address the issue of presumed azoospermia. So these are the patients where when you do your standard semen analysis, even when you're spinning down into a pellet, you're seeing 0 sperm. Some of these patients are going to go on to genuinely have 0 sperm, even with everything that we have available to us, including surgical sperm extractions. And for others, I was very excited to see this in the draft of the WHO 6th edition, where we are starting to see people realizing that there is a possibility of undetected sperm, even when we do a semen analysis with centrifugation. So we use the extended search protocol that's laid out in this paper, where we take that semen sample and use mini bilayer gradients to spin it down into our final preparation is a 100 to 200 microliter sample. And we take that sample and divide it into 5 microliter droplets in a 100 millimeter dish. So we flatten out the droplets, cover them with oil, and then search each droplet systematically so that if we can find small numbers of sperm, we can isolate them with our ICSI needle and collect them into small collection droplets. The same research group then developed the Sperm VD, which Xavier actually had a better picture of in his slides. So this is a sperm vitrification device. We load it with 1 microliter droplets of a 1-to-1 mixture of sperm wash and sperm freeze. And then we place the small numbers of motile sperm that we collected into these droplets with an ICSI needle. The whole device goes into a 2 ml sperm cryovial and gets plunged directly into liquid nitrogen. So this works really well for what it was designed for, which is the virtually azoospermic patients. And what we found was very quickly we were getting people coming to us or referred to us that had higher sperm counts. And by higher, I mean that I had to look very hard to find a picture in my microscope camera that had more than 1 sperm in the same field for this. So these are your 1,000 counts, 10,000 counts, 100,000. So these patients are still very much in that realm of we're concerned that we might not have enough sperm for ICSI. And yet we know that standard banking doesn't work well with such low counts. So until AI takes my place, the problem we had is that there's just a physical limit to how many sperm I can freeze at one time. So, you know, I can count them out one by one, but there was a certain amount of selection that's happening on my end that I really didn't want to be doing. My goal is to get as many sperm as possible into your ICSI dish so that you can do the selecting on the other end. I spoke with the lab that developed the sperm VDs, and their suggestion was, with counts like this, maybe we can take that little pellet and freeze droplets of the suspension without isolating the individual sperm. And I didn't love that because I really wanted a scenario where you can have some degree of presuming viability on the other end, because I only freeze motile sperm. So we know that 100% of the sperm that are frozen were viable before we froze them. And you're also going to bring over all this debris that you see in this image here as well if you're freezing the pellet. So that's when we started looking at Zymo. And my thought process with that was, if we have a way to take this, these very low counts and cohort just the progressively motile sperm in the samples, then, number one, that's all you need, right? You don't want me bringing over twitching sperm if we have progressive. You don't want dead sperm, you don't want the debris. They also have their data about decreased DNA fragmentation in the final preps that was very intriguing as to maybe it can be beneficial with this particular patient population. So I reached out to Zymo and we said, how low can you go? And they said, well, we don't really know. So we took all the cases that came into our lab where we saw a minimum of 1 motile sperm in the raw sample and a maximum of 1 million. So most of these samples have a maximum of 1 million concentration altogether. There's, I think, number 4 and number 27 here, a maximum of 1 million motile sperm. So none of these look like great samples when you look at them from the beginning. The only other criteria that we had was a minimum of a 1 ml semen sample, so that we put 850 in the Zymo and still had something to wash with the density gradient, just in case. And the decision as to which sample we used on the other end was really a matter of just comparing them side by side and seeing which one looked better. So the short answer here is that we got sperm for every single one of these. The first little group here, these are samples we were able to take right out of the Zymo and use them straight. We very quickly saw that some of these samples were getting back into that range of, we would have to do our whole extended search in order to find enough sperm to do ICSI. So we borrowed the conventional insemination spin. So we take that 0.5 ml sample you get out of your Zymo, and we spin it for 5 minutes at 300 G and take it down to 0.1. So we don't resuspend at that point. We use that final sample at 0.1. And then you can see the rest of these cases were, we were able to see detectable sperm just by looking at one droplet under the microscope. When we get down to this group over here, these samples, we did have to search through the entire 0.1 ml to collect enough sperm to do ICSI. But we did get them. This last group down here, these 3 were the only samples where we ended up using the gradient instead of the Zymo. And nothing stands out to me particularly about them. What happened was that we didn't see motile sperm on the other end, which seems like it is most likely some sort of laboratory factor, because I would have expected to see 0 sperm if they weren't moving through the Zymo. My suspicion is that not sure where I got lost.
Here we go. Into your presentation.
I'm not sure where you lost me.
Yeah.
So this last group here, we did not see motility, but we did see sperm come through the zygote. What we suspect is that we typically handle our sperm samples at room temperature, but the zygote protocol is 37 degrees. And we think that working with these poor quality samples, they weren't happy with that temperature fluctuation. So that's something we're going to be looking into making a change. But most of these samples we did freeze on the sperm VDs, but I do have some outcomes here of samples that I know were used for IVF, for ICSI. 3 of them were used fresh, and the rest of them were frozen first. And then these are some outcomes that we have. So I'm not sure that I can say that we moved the needle yet in terms of better outcomes for the patients, but it definitely dramatically improved the ease of getting sperm from the raw sample for these patients into the ICSI dish. I have a really great side-by-side here that I love. This is on the left is the gradient prep for the sample, and the right here is the zygote prep straight out of the zygote. Now, I think I'm just about out of time. I wanted to close with showing you the last scenario where we use single sperm freezing. I almost forget to start this video because nothing changes when I do that. So these are the samples with the undetectable motility. If you keep your eye on this little guy over here, there he goes. Okay. So these are the samples where, we do still have to do manually to find the rare motile sperm and freeze them for you. So, happy hunting, everyone. I look forward to taking your questions.
Thank you very much. That was really fascinating. I have a question to start you off. So when loading the sperm vitrification device, what is the maximum amount of time that I should spend looking for motile sperm before loading it? So what's the time frame?
So that's a great question because we don't use time as our factor. I think a really important part of how we search is that gradient prep, and the zygote does the same thing in that we use a really clean sample. So if I have a very dense debris field, I'll actually dilute it so that when I plate it in those droplets, it's very easy to see it, or I don't have cells on top of each other. And then we search from the beginning of the dish to the end of the dish. So the clearer the sample is, the quicker that is going to go, but we don't use how long as our factor. I'd say that a search can take us anywhere from 1 hour to 4 hours.
Wow.
Like I said, our favorite topic, most people's least favorite topic.
Yeah, quite possibly, yes. Thank you very much. I'm sure there'll be lots of questions later on as well, but for now, I'm going to move this along and introduce our next speaker. Debbie Montjean received her doctoral degree at the University of Paris and then completed a master's degree in physiology and physiopathology with a specialization in reproductive and developmental biology. Her PhD research project dealt with genetic and epigenetic anomalies associated with male infertility. Dr. Debbie Montjean worked during 9 years as a clinical and research embryologist in France, and she's currently working in an IVF laboratory as the lab director at Fertility Clinic in Canada.
Thank you very much, Kimberly, for this introduction. Can you see my slides? Yeah, probably.
Yes, we can.
Yeah, it's all good. I'm very excited to be there today, and thank you for pronouncing my name so nicely. It's rare. So my talk is entitled "À la recherche du spermatozoïde parfait," which in French means looking for the perfect sperm. So French is in French because French is my first language. And I'd like to thank the I3 initiative for having me again here today in this very interesting discussion. So I will firstly start with a fact that I'm sure that you will all agree on, is that as embryologists, we put a lot of effort in looking for the perfect sperm to perform the fertilization. So to pursue this goal in our lab, we took the decision to use microfluidics for all sperm prep for ICSI, because we do believe in microfluidics for the reason that were already discussed, so I wouldn't go into that. And then last year, we had access to artificial intelligence in our lab. So we have implemented SEED, that is an automated sperm selection system that that was mentioned earlier in our lab. And we were wondering whether SEED could help us to take sperm selection like a step forward, to have a better selection with SEED. So to combine SEED and Zymote, whether it will help us to increase our lab outcomes. So here's a short video on how SEED works. So basically, you just need to point the center of the field with the ICSI pipette, and then the system will scan the field and flag you the different sperm based on the different motility parameters, the different sperms that you could select, rank them from A to C, and from good to best quality sperm. So to assess whether SEED would help us to better select the sperm, we decided to design a small trial in our lab where we divided the oocyte cohorts that we had for ICSI into 2 arms. So we just split the cohorts. At the moment, we have more than 500 oocytes in our trial. 249 of them were injected with the traditional way, with the human eye. So Zymote was used for the sperm prep, and then the sperm that was selected for the ICSI was selected by the embryologist, and 257 of the oocytes seedling oocytes were injected using seed system. And then we tracked our lab outcomes based on Vienna consensus. We track these outcomes and we compare them based on the experience of the embryologist that did the ICSI. And in our lab, we also have time-lapse, which enable us to track all the morphokinetics parameters of the embryos that were derived from this oocyte injected in both groups. So here are the results of our study. If we start with the lab outcomes, we've looked at many outcomes, but I didn't put them all in the presentation just to make sure everybody could read the slides properly. So we've looked at the fertilization rate, cleavage, the day 2 development rate, day 2 embryo quality, day 3 embryo quality, blastocyst formation. And although there's a trend towards better results using SEED, that SEED is in orange here, and the traditional ICSI with the human eye is in blue. We saw a trend towards better results with using SEED, but we didn't see at the moment any significant difference, statistically significant difference. Then we looked at the same outcome based on the experience of the embryologist. So in our lab, we consider embryologists senior when they have more than 5 years of experience doing ICSI. So when a senior embryologist does the ICSI, we did not really see any difference in terms of fertilization rates, day 3 embryo development, and blastocyst rate. But there is clearly a trend toward better results using SEED when the embryologist who performed the ICSI is a junior embryologist, so less than 5 years ICSI experience. As I told you, we have time lapse, so we were able to annotate all the time points of the development, the embryo development, and we've divided the time points into 2 different groups. So the events that are related to the fertilization, namely the second polar body exclusion, the cytoplasmic wave, PN appearance, and PN disappearance. So all these were tracked. We didn't see any difference between the oocytes that were injected with sperm selected by seed as compared to the human selection. We did see a small difference in terms of second polar body exclusion. We saw that when seed is used, the second polar body is excluded earlier than when seed is not used. And again, for all the events that happen after, we didn't see any difference in terms of morphokinetics when seed is used. Only for the first cleavage, the time to obtain 2 cells is shorter when seed is used, but we, at the moment, we're currently not able to explain this difference. And exactly the same results were obtained for the embryonic cycle when seed is used. So the conclusion of our small trial is that although we didn't see better results when seed is used, this was a good news because seed gave rise to similar outcomes in embryo morphokinetics, which indicate that it's effective in sperm selection, at least as effective as a senior embryologist. Seed could be used for, it could be very useful in the presence of less experienced lab member. And of course, it does help standardizing sperm selection. So with that, I'd like to thank you for your attention and thank the team who performed this small trial in our lab. Be happy to take the questions if you have any. Thank you very much.
Thank you so much, Dr. Montjean, for the talk. I have one question. In your lab, what are the cutoffs for using Zymote for ICSI, IVF, and IUI, for example?
As for ICSI, we use Zymote on a daily basis for ICSI. Every time we would see a motile sperm on the macula, we go for the Zymote. We'll try our best to use the Zymote in every single case.
Yeah, thank you. And our next speaker, Dr. Zuzana Holubcová, is a lecturer and principal investigator at the Faculty of Medicine, Maastricht University. Her research interests cover multiple aspects of human oocyte and embryo development, including embryonic stem cell biology. Let's hear from Dr. Holubcová.
Hello, thanks for the introduction. It's my task and privilege to speak about molecular basis of fertilization process and what makes the perfect date between Mr. Sperm and Miss Egg. So the basic prerequisite for a successful date is to be at the right place at the right time. For ovulated egg, the place to meet her Mr. Charming is just the bar next door. But the sperm must take an epic journey to get to fertilization site. Passing through female reproductive tract, it has to overcome mechanical, chemical, and immunological barriers. So only the best individuals would survive this big sperm race and reach the fertilization site. Good manners dictate that ladies should be waited for. So the sperm take the opportunity to chill out. They adhere to the epithelial cells in the oviduct, making a sperm reservoir. This deposition extends sperm viability until the oocyte arrives. It's estimated that human female reproductive tract is capable of keeping sperm fertilization competent for about a week. Presence of the egg activates the male gamete, which is deposited in sperm storage. To get out of the sperm of this deposition site and penetrate egg vestments, the sperm motility pattern shifts from rotational flagellar movement to intense asymmetric beating. This transformation is called hyperactivation, and along with capacitation renders the sperm competent to fertilize the egg. The change from basal to hyperactivated motility is triggered by a rapid rise of intracellular calcium, resulting from an influx of calcium across the sperm plasma membrane. Calcium serves as a second messenger in cell signaling and directly enhances dynamics of cytoskeleton components in sperm flagellum. PET clamp technique, which you can see here, enables to study ion movements across membrane. And these recordings help to uncover a central role of Cat-Sperm channel in calcium signaling. Cat-Sperm is a pH-sensitive ion channel. Its expression is testis-specific and location is confined to surface of the sperm tail. Genetic evidence shows that the function of Cat-Sperm channel protein is indispensable for male fertility because knockout mice are sterile despite having normal sperm count and morphology. But their spermatozoa exhibit impaired motility and fail to penetrate zona pellucida. In humans, genetic lesions and altered expression profiles of Cat-Sperm genes have been clinically linked to astenospermia and male infertility. Opening of this multi-protein complex allows rapid calcium influx and rise of intracellular calcium, leading to sperm hyperactivation. A conformational change of the blocker molecule, which you can see here at the pore of the Cat-Sperm channel, requires a rise of intracellular pH, which provides a molecular explanation for hyperactivation being primed by alkalic environment. Two simultaneously published top-ranked studies revealed an unprecedented role of sex hormone progesterone as an hyperactivation-inducing factor. This female steroid hormone, secreted by cumulus cells that surround the oocyte after ovulation, it's well known as a chemoattractant for mammalian spermatozoa. Nanomole concentrations of progesterone has been shown to dramatically potentiate hyperactivated flagellum movement of human spermatozoa in alkaline environment. Surprisingly, progesterone-induced hyperactivation doesn't involve slow canonical pathway typical for steroid hormones, which bind to nuclear receptor and alter gene expression. That would be a very long task. This study, published in 2016, showed that action of progesterone is non-genomic. It's not based on the nuclear receptor. It's on Cat-Sperm. It's mediated by this molecule called alpha-beta hydrolase domain containing protein, which after progesterone binding hydrolyzes this substance called which is endocannabinoid called 2-arachidonyl glycerol, and this compound is normally it's highly enriched in the sperm membrane. And after this action, after the hydrolysis removes arachidonyl glycerol from sperm membrane, leads to full opening of Cat-Sperm, which triggers sperm hyperactivation. However, it's not just progesterone which can interact with Cat-Sperm. It's a promiscuous channel, and number of compounds can interact with this channel. We've reviewed literature. Sorry, this is moving. Yeah, we reviewed literature and found that both testosterone and hydrocortisone, the steroid hormones, at physiological concentrations are capable to replace progesterone from its binding site on the hydrolase molecule, and so sort of act as antagonists of progesterone on this receptor, which could explain why elevated levels of these steroid hormones are adversely negatively affecting female fertility. The predominant expression of Cat-Sperm on sperms makes it very attractive substrate target for modulation of male fertility. Plant triterpenoids, triterpenoid lupel was identified as a potent inhibitor of hyperactivation in incapacitated spermatozoa. It's again competing progesterone from its binding site. Importantly, a range of structurally diverse molecules belonging to environmental pollutants, endocrine disruptors, odorants, and other harmful chemicals can also interact with cat sperm and possibly desensitize this channel to progesterone action. So in fact, interfere with its function and impair its functional competence. Research of fertilization in humans is hindered by many legal and ethical restrictions, but time-lapse microscopy provides opportunity to monitor interaction of human gametes used for fertility treatment. Here we could see how spermatozoa draws through the matrix of zona pellucida and gets in physical contact with the egg. After reaching perivitellin space, the spermatozoa adhere to membrane protrusions known as microvilli, as you can see here, which are densely covering the surface of the oocyte. The initial approach is followed by steps of molecular recognition, membrane tethering, and finally fusion of plasma membrane. The molecular mechanisms, as I said, investigating fertilization in humans is very problematic. So we take advantage of very popular mouse model. So this molecular complex dialogue has been investigated in mice. So far, from that, there were a great number of genes implicated in the process of fertilization, but only a few of them have been proven essential for fertilization in mouse model, which means that mice in which the gene was depleting show a sex-specific defect of fertilization, despite having normal mating behavior, gamete number, and morphology. So they are basically sterile. That's the basic requirement for gene to be proven essential in the process. The first gene, which was shown to be essential for gamete interaction, is CD9. It's a membrane protein localized on the oolemma at the area of microvilli, and it's required for normal microvilli morphology and distribution. It's interesting that CD9 containing vesicles can not only rescue eggs which lack CD9, but they can also transport the part of CD9 positive membrane to sperm membrane before fertilization, which means that it's not only a rearrangement of oolemma, but also sperm membrane, which is required for steps of fertilization or gamete interaction. Besides CD9, another tetraspanin, which is a surface molecule called CD81, has been implicated in the fertilization process. It's also expressed on oocyte surface, and it associates with CD9. It's also incorporated into microvesicles, but it's a different population than CD9 positive vesicles. And its depletion inhibits gamete fusion in vitro, but the mice deficient phenotype is not sterile, but just subfertile, but can be rescued by CD9, which suggests that there might be a synergic effect between CD9 and CD81. So it's assumed that CD81 together with CD9 contributes to arrangement of oolemma architecture and also sperm membrane rearrangement in preparation for for gamete adhesion and or fusion. The first identified sperm antigen involved in fertilization process was isomo-1. It's named after Japanese shrine of marriage. It's expressed on inner acrosomal membrane, and thus it's undetectable on the surface of freshly ejaculated spermatozoa. But at the time of acrosome reaction, isomer becomes exposed and diffuses from acrosomal cap to equatorial region where the sperm of the sperm head, where the gamete fusion is initiated. Both isomer 1 and CD9 are enriched in adhesion area, so the expectation was that these might be interacting factors. But binding assay showed that there is no physical interaction between the two molecules. So what's that receptor for isomer on X surface? This receptor was identified in 2014, and it got the name Juno after ancient Roman goddess of marriage and childbirth. So Juno is a isomer complementary receptor. Juno deficient cells do not fuse with spermatozoa. Interestingly, ectopic expression of fluorescently tagged Juno and isomer is capable to induce very firm adhesion. You will see that these cells which were transfected with these two proteins, Juno is in red and isomer is in green, they are really firmly tethered, but they do not unite, meaning that these two proteins, they do bind, they pair, but they lack fusogenic activity. They are not, there must be another factor involved in fusion. So this led the researchers to propose a model in which isomer and Juno binds, and this binding induces accumulation of CD9 and possibly also CD81 and possibly other factors at adhesion site, and thus promoting clustering of other membrane proteins, basically cell fusion machinery, which then helps to merge membranes of the two gametes. In recent years, the introduction of CRISPR technology and also other gene targeting experiments revealed other sperm expressed proteins which are involved in fertilization process, because the mutations and or depletion causes failure of gamete fusion. But their precise role in molecular recognition is still unclear. Very recently was this paper came out where researcher used, instead of investigating fertilization itself, they used a decoy bead with proteins, with protein ligands, which was mimicking the egg, and they were assessing sperm binding. And using this approach, they were able to identify a protein which they called Maya, after a Roman or ancient Roman nymph, which was related to Juno. Maya is a protein which is expressed on oolemma of unfertilized human oocytes in close association with Juno. And here you can see also electron microscopy showing that they really they not colocalize, but they have the very close association. Transgene expression of co-expression of Maya and Juno led the sperm binding to primary cultured cells. So these cells, not the egg, just primary cell, when it was expressing these two molecules, was capable to bind and internalize sperm head, which means it shows Maya's place in fusion machinery. The researchers proposed this model in which isomer first binds to Juno, which causes initial adhesion. Then dimerization of isomer causes conformational change, flipping of Juno, which later leads to Juno shedding as a part of polyspermic block mechanism. And the emptied pocket in isomer structure is then filled with Maya, which by conformational change is capable to really tether the neighboring membranes, but still we lack the fusogen. So we got now more complete picture of multiple factors which are involved in the process, but still what's that mysterious fusogen? Early this year, this study came out, which indicated that the fusogenic protein might be actually that old good isomer 1, that sperm expressed protein, which is initially binding to Juno. They showed that this protein has two domains. One of them is responsible for binding to Juno, and another one is capable to induce unilateral fusion of primary cells. Here you will see the two cells expressing this protein, and they are really fusing. So they proposed alternative but better model, more complete model, in which the fusogenic activity of isomer follows transient adhesion mediated by Juno and isomer. And later, probably the Maya replaces Juno, allowing very close tethering of membranes, which is a prerequisite for isomer-driven fusion. So from what I said, I think it's clear that not just like with human dating, there is no, this applies to gamete as well, there is no say it all, chat up line, just for gametes, just simple lock key interaction, like just isomer and fusion happens. But instead, there is a very complex molecular dialogue involved in this process. What happens if this process doesn't work, or if there might be some mutations of receptors, altered expression of either receptors or signaling pathway components, or there might be some interfering agents. So in that case, the date won't be perfect and won't result in successful fertilization. And in our real world, it might be underlying idiopathic infertility and IVF observed fertilization failure. So why it's important to study these processes? I think better understanding of basics of human fertilization would lead to improved diagnostics and informed consulting, better improvement of gamete culture and in vitro fertilization conditions, better biomarkers for sperm selection, and possibly also for this knowledge could be generated by basic science, could be good for development of a new generation of short-acting non-hormonal contraception. For sake of time, I didn't cover topics like polyspermy block and egg activation, but if you are interested in the molecular basis of this process, check our publication from 2020. And thank you for your attention, and I'm happy to take your questions.
Thank you very much, Zuzana, for a really interesting presentation. Thank you so much. The first question that I would like to ask you is what you mentioned about the CatSper channel. I find that really fascinating. Do you have an estimate on the amount of patients that could be affected by a non-functioning CatSper channel?
I'm not sure how many, but definitely there are polymorphism and altered expression profiles linked to CatSper. But how many patients are actually affected? It would be just guessing. I do not have clue.
Okay, yeah, thank you very much. And with that, we would like to start the question and answer session of our webinar. And the first question that I would like to ask is to our panelists, so Professor Palermo and Caroline. A lot of questions have been coming in about the Zymo device and how it's used and what it's compared to. And most of the studies out there compare the Zymo device to density gradient preparation. Have you compared the Zymo device and its results to SwimUp results?
Should I answer? Hi, Gianpiero. So
yes,
we don't use SwimUp anymore as much in our center at all. So it's done very, very rarely because of the higher incidence of malfactor patient, of subtle malfactor, to say better. So we compared only with the density gradient we were using. I think for some people that have done this experience, the SWIMAP is comparable to the zymote, because if you have SWIMAP, you have outstanding sperm parameter. As everybody knows, when you use a SWIMAP, that's what you do, and you have a very high motility rate starting with. So you will be able to get sperm with high motility, high concentration of sperm with mostly motile with a lower DNA fragmentation rate, comparable to the zymote.
Caroline, would you agree with that?
Yes, I definitely would agree. We do use SwimUp when we have, a non-viscous sample with good counts and good motility, and it actually, it works very well as well. I think we use zymote now more for samples that we would have actually used isolate for in the past. They may not be, quite meeting the numbers for the SwimUp samples that we typically do. But they seem to work well on the zymote. So outcomes, I think it behaves very similarly to SwimUp samples. When you get the sample, I don't know that outcomes in terms of FERT or euploidy or anything would be better. We found them very comparable. It's just the samples that we choose whether to process them via SwimUp or zymote.
We received another question from the audience that kind of ties up into this conversation. As you, Dr. McCaffrey, mentioned earlier, you said that morphology does not play a big part in the decision-making. And could you and Dr. Palermo comment on whether you consider grade of motility an important factor? So in other words, to what extent does the speed of motility matter in this decision-making?
If I may, well, I want to kind of distinguish a little bit. When for us, very important, the morphology. The reason why indirectly the morphology is important, because you distinguish between a sperm parameter or spermatozoa. There are certain parameters, spermatozoa, that are functional. If you have an abnormal small acrosome, you will have a problem with fertilization. And that was the issue when standard IVF was the only thing known. There were, where I started in Belgium, and when I came to New York, the fertilization rate with this case was, there was the chance of fertilization failure about 40% of all the patients undergoing standard in vitro insemination. The problem was, although there was enough spermatozoa, was the morphology, but the morphology, according to Kruger, looking at the acrosome. So I think the morphology is important to that aspect. Why, of course, if you adopt ICSI, morphology is not important, motility is not important, the concentration is negligible. But anyway, so the parameter, the question was, the parameter related to what? The motility related to what?
Just the speed of motility.
I mean, motility is key because the motility will tell you if the sperm is alive. So if you're searching for hours, like we heard the presentation before, where you search for many hours to identify the spermatozoa, the motility only important because it tells you that the sperm is alive. But if you have a sperm that is alive and is immotile, is as good. My humble opinion.
Yeah. I'll ask another question then as well. Would you say that zymote could be used for frozen sperm? So, for example, donor sperm?
It's a question for me?
For everyone.
For anybody? Yeah, I mean, if anybody want to take it.
May I answer this question?
Yeah.
Yeah. Yes, we use it actually for donor sperm. As long as we have motility, when we assess sperm motility and sperm count in the macula, we would use the zymote. So yeah, it's very efficient on frozen sperm.
Can I follow up on that? So when you thaw the sperm, do you get rid of the cryoprotectant first, or do you put it straight into the zymote?
You need to dilute a little bit the cryoprotectant, at least half. Yeah.
Yeah.
because it's going to stay there, I mean, in the device for half an hour. So better to dilute the cryopregnancy first.
I think it may clog the pore too, I think,
Yeah.
because it's more viscous.
Definitely. It's
Yeah.
advised by Simon.
No, yeah, I don't know. We were on the pilot, we did the pilot study with Zam before they even develop any criteria. So we've been using with frozen sperm, and it works well, as you mentioned, as long as there is motility. What we noticed, though, if we cryopreserve a sample processed by Zymote, so we get higher proportion of motile spermatozoa, so the survival rate, the post-storm motility is amazing. That's our finding in a small series.
Okay. And would you recommend to only use that on the non-prepared semen sample that was frozen, or would you even use it on prepared semen sample that was frozen?
What do you mean by prepared? Do you mean like
Process?
washed?
You mean process?
Yeah, processed.
Washed?
Yeah.
Yeah, both.
Both.
Yeah.
Okay.
You mean,
I
but
have
the
a
whole
question.
idea is to use Zymote with less manipulation possible to avoid exposure to centrifugation, to reduce the exposure to the free radicals. So that's the idea of using Zymote. Yeah.
Yeah, we have some patients who insist on having Zymo used on the sample, and every now and then we will say, well, it's already been processed and
Oh,
then
I see.
frozen, but they still insist on it. So
Sure.
yeah. I have a question for Chaya from Eloisa's Papayanis. Vitrification is still considered experimental by the latest World Health Organization guidance. Do you use vitrification for fertility preservation male patients as well? And if so, how good is the survivability during the test thaw cases?
So I can tell you that we don't use it for normal samples. We use it for fertility preservation when they meet all the other criteria that we use it for. So if they are, cryptospermic or virtually isospermic, our scenario where we have very little sperm, we're worried that won't survive standard thawing. So we have used it for trans patients and pre-chemotherapy if they meet those other criteria. I don't think we've been doing this for about 4 years. I don't think we've thawed any of those fertility preservation samples yet.
Okay, yeah, because you have a low number of sperm, you obviously wouldn't be able to do a test thaw, right?
Right, we never do test thaws on these.
Yeah, of course, that makes sense. I have another question for Debbie. When you use the AI selected sperm, so the SID sperm, do you have any data yet on birth outcomes?
Actually, we do. Yeah. They are obviously birth around the world because we're not the only clinic using the SID system. So they birth already that were reported. For internal study, we're still collecting data about cumulative pregnancy, about the diploidy of the embryos, and we already have birth in each arm. So yeah, it works.
Yeah, very good. And I would just move on to Zuzana, because you mentioned about male contraceptives and Catspur being a possible target for male contraceptives. Do you really think that can happen? And do you know of anything that's in the works?
I'm aware of a company which was interested in Lupel, that triterpenoid, which could replace, compete out progesterone. But honestly speaking, I see this might be very problematic because the question is reliability. This contraception could be rare because there might be just one sperm which could escape this negative regulation, and then the customer would be very unhappy. So not sure. I'm not super optimistic in this, to be honest.
Yeah, that's very true. I mean, you would have to disable lots of sperm and all of them in order for it to work.
I'm much more optimistic or excited by research of that interaction between gametes, because that could be unisex contraception, theoretically. But yeah, at the moment we are just uncovering basics. So, and that's the prerequisite for pharmaceutical industry. So, but yeah, honestly speaking, I don't think this is gonna happen any soon. Or I wouldn't rely on it.
I have another question for Zuzana. It's coming from David Morrell from UK. Is the hyperactivation a critical function for release of sperm from epithelia, like the oviduct, penetration of the cumulus mass, penetration of the zona, or all of these?
Yes, exactly. Yeah. It's this change of movement is critical. It's that swimming, ordinary swimming basal motility wouldn't be sufficient to get through really viscous, dense environment. Detachment from the sperm reservoir, and getting through all cumulus, getting through zona. So yeah, it's critical function.
Sorry, Neringa, I think I lost you there for a second.
Sorry.
One question I would like to ask our entire panel, because it's a question that keeps coming up, and I think it's one of the main questions that I need to answer whenever anyone asks me about the Zymo device. And Chaya, I think you don't have to answer this one because I think you showed an excellent data set in your talk, but people keep asking about cutoff values. And I know, Chaya, you don't have any cutoff values. You demonstrated that quite impressively. But does anyone else have a cutoff value in their clinic, or is it just a trial and error and see if it works?
When you say cutoff value, related to what? I'm not sure I understood.
So for example, do you have a cutoff value? So when a patient comes in and let's say the patient only has a concentration of 0.1 million sperm and maybe 5% motility, do you from the start say no Zymo preparation for you, or do you say we'll give it a try?
Yeah, we use Zymo regularly in our embryology laboratory, but for this particular case, we do it in a separate laboratory, which is the andrology lab, where my andrologists are trained for ICSI. We have different allocation of the personnel at Cornell. So what we do as long as we can retrieve some spermatozoa, we tell the patient and the physician, the REI, sorry, we're going to try, but we're not sure. I prepare the man to produce at least 2 or 3 samples. I want a very short abstinence. And then we'll process through the zymote. Then even if we don't see spermatozoa on the other end of the zymote, above the membrane, we make ICSI dishes at 5 or 8 microliter droplet, and we look at an inverted microscope. If we found some spermatozoa there, we will use for ICSI. So really it's unit of sperm. It's very hard to establish a concentration. But we'll use, as long as there is one sperm, we'll use it.
Yeah, same as Chaya then, right?
I do have to say though that the one thing we haven't tried yet is if I've seen 0 motile.
0 motile won't go to the zymote, obviously.
We have the same policy here. And what we need to keep in mind is that not all the sample is going in the zymote, so that you can try, because it's only 0.8 that goes in the zymote. So if really you end up finding no sperm, then you can use the other fraction of sperm to do another sperm prep. So it's, to me, it's worth trying. Yeah,
in that case, we use a centrifugation or to reduce, but if you get too crowded, then I can use more than one zymote chamber. Okay. It's costly, but then you have to work with the surface of the membrane and the concentration of the sperm.
Yeah, that's a good point. And another question to Zuzana from Shakira Musa from Johannesburg. Can we then treat severe actinozoospermic patients with progesterone supplementation?
It depends whether that osteospermy is caused by cut sperm mutation. If there is a defect in a channel, I wouldn't expect progesterone to be effective, because it's potentiates, it opens the channel, but it has to have normal structure, or close to normal expression. So if there is lack of this protein, I doubt progesterone would help. So it's, yeah, don't think this is.
If I could make a comment, I used progesterone in my first paper when I was doing subzonal injection. It helps the process of capacitation of the spermatozoa. So if you have non-motile sperm, they are alive, you facilitate the process of capacitation with progesterone. Really, it works on the membrane, but it won't resume the motility, for example. And most of the problem we have with motility, when there is motility, the problem with the cilia dyskinesis, primary cilia dyskinesis. Yeah, that's what the recurrent phenomenon we have.
Yeah, very interesting. One final question to Chaya. Have you considered using any other vitrification devices other than the sperm vitrification device? So have you looked at using normal straws, for example?
This is kind of a dream of mine. As I said, I think it works very well for what it was designed for, and I love it for single sperm. I wonder if for patients with those, 10,000 sperm counts that don't freeze well in a vial, but it's way more than I can freeze individually, if there's not some bridge method that we could be looking at.
Yeah, that'd be really interesting. Definitely. Right. I would like to thank everyone who took part in this webinar. I do think we have had great presentations and great talks. I would love to thank my co-host Neringa and of course our expert panelists, Professor Palermo and Dr. Caroline McCaffrey. And obviously, I won't forget our team from I3. Thank you so much to the organizing committee, namely Jack Cohen, Peter Nagy, Mary Cervantes, Thomas Elliott, Fren Farley, Giles Palmer, and Violetta Sora.
Thank you everyone so much. And this webinar was kindly sponsored by Zymat Fertility. Thank you, Zymat. If anyone wishes to support I3 or send interesting topics for future webinars, we would be really grateful. Our next session is on the 28th of November, and it's called Self-Control, and it's all about cell division and origins of genetic errors in human embryos. Hope to see you there.
Thank you. Bye-bye.
Bye-bye.
Bye.